Good day, everybody listening to the talk here from sunny South Africa. And a big shout out to Protochips for giving us this opportunity to present a few slides on some of our recently published work in applied catalysis A, on some reduction behavior of silica-promoted iron oxides using in-situ TEM. So precipitated iron oxides are frequently used catalyst precursors in industry for the low temperature Fischer-Tropsch process. And as is common with many catalyst systems, structural promoters are employed, in this case silica, to promote a specific structure of the material. And in this case, it's to promote the formation of small crystallites of the active phases. In this case, it is iron, and it also aids in improving the mechanical integrity of the catalyst as it is used within a reactor. It's a little bit of a double-edged sword, the promotion with silica, because if you add a little bit too much silica, it could cause an inhibition of these oxide precursors to reduce fully towards active phases. That inadvertently could then affect the catalytic activity of the catalyst. So it's important to really find the appropriate balance of the amount of silica to use. The reason why this happens isn't entirely understood. From other non-direct characterization techniques, it seems to point towards the formation of iron silicate phases during reduction. But it was our aim in this study to try and look at this in a little bit more detail by combining the use of other in-situ techniques such as in-situ XRD with the use of in-situ TEM to get a complete picture of the mechanism of reduction of these catalyst precursors. So the first port of call when analyzing a catalyst is typically using some kind of bulk characterization technique to look at how this material reduces. In this case, here on the right-hand side, you can see examples of temperature program production studies done on this catalyst system. And on the left-hand side, you can see the results of an in-situ XRD analysis of the reduction behavior in hydrogen. If you add a little bit of Rietveld refinement to your XRD data, you can actually get quite a nice amount of information about what type of phases are present, what the relative abundance of the phases, as well as the mean crystallite diameter of these phases are at each step of the reduction process. Now, with all of this information, and it is already a very, very good description of what's happening, there is some aspects missing. What we're missing is a full description of how the morphology changes with reduction. We also don't have any idea really where the phase forming regions are. We don't have an idea of the crystallite size range, and very importantly in this case, the distribution of the silica as the precursor reduces towards the active phases. So to study this in a little bit more detail, we decided to use the aberration-corrected microscope housed at the University of Manchester in combination with a Protochips atmosphere TEM environmental gas holder. We took the powders, we dispersed and sonicated them in ethanol, and then put a very, very small drop on this heater MEMS chip, and then left it overnight to dry. Came back the next morning and we assembled the chip into the holder as indicated by this figure at the bottom left, and then put it into the microscope for successive reduction steps to study the dynamic changes that occur on several sites of interest as we reduce at temperatures up to about 750 degrees. The question, and one of the most important aspects of why we ended up using an in-situ system, was that iron really readily reduces in an ambient oxygen environment. It oxidizes to form surface layers when you just expose it to air. And we really tried to avoid that in this study because this could complicate the interpretations that you make from successive reduction steps quite a lot. So what we did is we used the in-situ XRD data as a guide to know in what temperature regimes we needed to go and look at to study the morphological changes as well as the distribution of silica as this system is reduced. So we did that, as I said previously, from room temperature up to 750 degrees. We selected several sites of interest, and then we followed the changes of these several sites of interest as we reduced at these different temperatures. So here's an example of an unpromoted iron oxide precursor and the several steps of how the morphology changes with different reduction steps as we go along. And you can see that we're looking at the same area the whole time. It's correlated. You don't have to try and interpret findings in between different areas. You're looking at the same area the whole time. And over there, you can see a nice depiction of how the morphology changes over time. Of course, you can do a lot more than that. So what we did is, in this case, on the left-hand side here, you can see the morphological changes of the promoted, silica-promoted iron oxide precursors at or after different stages of reduction. We also could then go and have a look at using, for example, selected area electron diffraction, how the phase composition in that area changes over time. To do that, we actually, in between every step, completely removed hydrogen from the cell by using the turbomolecular pump connected to the system, and then studied each site of interest after every reduction step using diffraction. We used spectroscopy, we used different imaging modes, different imaging techniques to get that. So on the right-hand side, you can see here is the successive selected area electron diffraction patterns obtained from this very same area as the reduction went along. And then by doing rotational averaging of these diffraction patterns, you can then, in a very similar way, generate electron diffraction spectra that you can go and use for phase identification in that area at or after the various reduction steps. So now we've got a better understanding of how the morphology changes. We've got a better understanding of the crystallite size ranges. And we've got some idea of these silicates forming in that area because we'd identified them from the electron diffraction data at the higher temperature end of the reduction. We still needed to get information about the distribution of silica or silicon and then also phase forming regions. So what we did is we used a technique called hollow cone diffractive imaging to put select a interplanar spacing of one of these phases. And the example I'm showing here is for magnetite, where you would use an objective aperture and process a tilted incident beam in 360 degrees to then give to you the positions of those phases exclusively. And then you can generate a stack of images, which you can then process to give you the distribution of or the positions of these phases that you're specifically looking at. What you can then do is here is a hollow cone image of that very same area after 450 degrees reduction. Where you can see the phase forming regions for magnetite. Over here, the bright regions are the areas that are very well crystalline in magnetite crystal structure. You can then correlate that to quantitative Eales compositional mapping of that same area. Over here, you can see this is a map of the oxygen or iron divided by oxygen concentration. You can see areas where you have got a concentration of iron is the likelihood for the formation of magnetite to occur. We could also, even though the silicon nitride window contains silicon, we could map the distribution of silicon to a certain degree by assuming that the silicon to nitrogen ratio of the window remains the same. And using that, you can then deconvolute the signal above this background silicon signal and get an idea of how the silicon actually diffuses away from iron-rich zones during the reduction process. And of course, you can link this all back to morphology. You've got everything that you need in order to develop the mechanism or to understand the mechanism of reduction a lot better. Another tip that a person can use to try and extract some contrast or additional information from EELS data in this case is also to use EELS fine structure mapping. In this case, we could map the oxides by using the little pre-edge of the iron oxide phases that have formed. So in conclusion, in-situ TM was really a very crucial and critical for us to be able to understand this reduction mechanism almost completely, if I can put it like that. It allowed us to collect complementary data sets at each point of the reduction stage, and then also correlate that to in-situ techniques, such as, for example, in-situ XRD. We got all the information, morphological changes, phase evolution, the distribution and movement of elements right down to the nanoscale. And with this knowledge, we could really understand the mechanism a lot better. And future studies of developing or improving on these silica-promoted iron oxides can then use this knowledge to fine-tune the amount of silica to add, as well as to develop the reduction strategy they should follow to activate these catalysts. And with that, I invite everybody to go and read the paper, which I hope will be placed as a link at the bottom of this video. And I would like to acknowledge the National Research Foundation and the Department of Science and Innovation of South Africa for their financial support, as well as the University of Manchester, a collaborator on this paper, as well as Sasol, also a collaborator, and once again, Protochips for this opportunity. Thank you very much.